hope everybody is well. I'm going to continue this lecture on chapter 2. We're going to finish very quickly probabilities. And after that, we're going to go into pedigrees and transmission genetics in humans. Now, last time we talked about the chi-square analysis, testing observed experimental numbers with theoretical numbers, and now we're going to quickly go through the product rule, sum rule, the conditional probability, which is something that I don't want to insist too much on. It's not that difficult. It's very well explained in the book, and you've probably seen it before. And then I'll say a few more words on binomial probability to remind you a little bit how that's done. Now, the product rule is also called the multiplication rule. That has to do with the probability of two or more independent events. That is to say, it's a probability that describes the word and. If you have one event and another event, the probability of both events occurring is calculated by multiplying the probability of each event. For example, the probability of having 3 heads in a row with a coin, each event being 1/2 is 1/2 times 1/2 times 1/2, which is 12.5%. So this is fairly simple. I'm sure you've seen it before, and this applies to different events that can have different probabilities. You can have an event that has a probability of 10%, another one 60%, etc., and just multiply the different events. But keep in mind, this is describing the necessity of both events occurring, both independent events occurring at this in sequence, and so it describes the and correlation. The sum rule, however, also called the addition rule, describes the relationship or. So if you have an occurrence that is independent of another and mutually exclusive, ask what are the two events occurring, what is one or the other occurring, right? It's the joint probability of any two or more mutually exclusive events. Then it's, for example, probability of getting 3 heads, as we had in the previous slide, or getting an even number with a cube, which is a probability, of course, of 1/2. So it's 1/8 plus 1/2. So the sum rule is you just add the two, and it's 62.5%. So those are very, very simple notions, I think. Now, the conditional probability, we're not going to go really over this because, in fact, in the book, it's explained very scantily. I just want to give you a flavor of what it is. It's actually, once you have a result, asking what's the probability of the events that produce the result. So in other words, if you see a progeny, say with one affected individual, what is the probability of having one parent being heterozygous, for example? Here's actually an example with the peas that we talked about last time. If you have 2 heterozygous crosses, and you between yellow heterozygous plants, so there you have, as you know, 3 out of 4 of the offspring will be yellow, and 1 of the peas will be homozygous wild type, and 2 will be heterozygous. So if you know that your seed is yellow, 2/3 in 2/3 of the time you'll have a heterozygous seed, and 1/3 of the time you'll have a homozygous wild type. So knowing that your seed is yellow, then you can say, well, if the parents are heterozygous, the chance of this seed being homozygous is 30%, 1/3. So there's a lot more regarding conditional probability. This is really the very beginning of it, and in fact, in pedigrees, this involves quite complex calculations. So, but I just want you to have a vague idea what this is. I'm not going to insist on that. What I want to talk a bit more about is binomial probability. So this involves the analysis of the likelihood of a series of events for which there are different outcomes and for which the probability. Now, an example that I'm going to give you at the next slide is, for example, what is the probability if you have 3 kids to have 3 boys, 3 girls, or 2 of one and 1 of the other? So this is predicting the probability of a series of events, 3 different children in a family, and what are the different outcomes, boys or girls. So for that, you need binomial probability to answer that, and you need to formulate the probability of each event first of all, and then you need to develop the number of events and giving you the probability for each sequence as a binomial expansion in the form of p plus q to the power n. P is the probability of one of the events, and Q is the probability of the alternative events. They do not have to be the same. In the case of having boys or girls, it's the same, it's 1/2. But as we're going to see, we're going to look at another example of a heterozygous cross. They can be different. But you need to be able to develop this binomial to the power n in order to have all the different events as terms that then you can use to calculate the probability of each succession of event independently. So here's the first example, and I think you're going to understand what this is about. So you have a family with 3 children. Predict the proportions with each possible combination of boys and girls if you have 3 children. So as I said, P and Q are the same here. They're 1/2, and so you know the probabilities. So what we're interested in is really 3 events, 3 children. So the expression is p plus q cubed to the power 3. So this is really the mathematical expansion development of this expression. Pq plus 3p squared q plus 3pq squared plus q cubed. And this is really what you need to know how to develop, and we're going to talk about this in a second. But before I do that, I want to make 2 comments on this expression. This expression, by definition, will be equal to 1. That is, you are expressing all the possible events that can occur in this setting. You have all the possibilities of boys and girls if you have 3 kids. If P is the probability of a boy, P cube is the probability of having 3 boys. 3p squared q is the probability of having 2 boys and 1 girl. P squared is probability of having 1 boy and 2 girls, and q cube is the probability of having 3 girls. So these are all the possibilities, and therefore this expression is equal 1, and each term that constitute that expression that are added together to add up to 1, each term is actually one possible outcome among all the outcomes possible. Like I said, 3 boys, 2 boys and a girl, etc. So what's the probability of having 1 boy and 2 girls? Well, it's 3 times 0.5 times 0.25. That would be the probability. So now let's talk about how you actually develop this, because you might have you might have to refresh your memory about that. So you're asking for 3 events. So it's p plus q cube, as I said, where p equals q equals 1/2. Those are the probabilities. In order to know how to do this, and no matter what the power, I mean, you can do it if you have 8 kids, okay? You see you have coefficients in this expression, 1, 3, 3, 1. These are called the Pascal coefficients, and they will be the coefficients for every term part of this expression. Once again, 1, 3, 3, 1. How do you get these coefficients? You look at this table, which is the table showing the binomial coefficient for the number of events shown here on the vertical axis. In our case, it was 3 in the previous slide. And you see there you have your coefficients here. Okay, 1, 3, 3, 1. So this is where I took my coefficient that I showed you in the previous slide, 1, 3, 3, 1. And you have 4 terms in that case, 1, 2, 3, 4, okay? And the next step is to develop the powers of P and Q for each expression. You do that by starting with P and give it the power of the number of events that you're analyzing. In that case, is 3 events. So it's P cube in our case, because we have 3 children. And then the power of Q will be the remainder of the number 3. So in other words, the powers of P and Q, if you add these two, they have to be equals to 3 for each term. So since you already have 3 here, the power of Q in this term will be 0. And Q to the power 0 is 1. So this, you can take it out of the term and it gives you P cube. Now, go to the next term. The Pascal coefficient is 3. You lower the power of P by 1, so it's P squared. And since I told you you need the powers of P and Q to be 3, that gives you Q to the power 1, which is Q. So this is how we get the term 3P squared Q, which is written here. The next term, you lower the power of P by 1, so it's 3P, and that gives you Q squared, because 1 plus 2 equals 3. And the Pascal coefficient is again 3, so it's 3P Q squared, which is this term here, the third term. And again, and for with the same idea here for the last term, it's 1Q cube. Not written here, P to the power 0, which is 1. Once again, the sum of all events is 1. That's what you see here in this equation. All the possibilities that you can have with 3 children of number of boys and number of girls. So once again, you can actually calculate, give a number, a percent, in, for example, like I said before, 1 boy and 2 girls. So that's 3 times 0.5 times 0.25. So here's the Pascal triangle. This is a mathematician of the French mathematician of the 17th century who sort of developed these formulas. And no matter what the number of events, like if you're thinking about the previous problem with thinking with 6 kids, well, here are the coefficients, and you'll have 64 number total number of combinations. But here are the different 1, 2, 3, 4, 5, 6, 7 number of events occurring. Okay, so this would be 6 boys, 5 boys and 1 girl, etc. Now let's look at another case, another example that is slightly more complicated, but you'll see now that you've seen what you've seen, it's not going to be too difficult. And that's using a cross that we talked about last time. When you self-fertilize a heterozygous pea plant, for example, for the yellow seed phenotype, and you remember that, as I just told you, in fact, 5 minutes ago, 3 out of 4 will have a yellow seed, meaning receiving a wild type allele dominant, and a fourth will have green seeds, small g, small g. So the probability of a yellow pea is 3/4, and the probability of a green pea is 1/4. So in that case, P and Q are different, and you'll see it's going to be exactly the same idea. So this is essentially justifying what I just told you, that the probability of a yellow seed is 3/4 and the probability of a green seed is 1/4. And we're going to look at what happens if we look at pods with 6 peas. So if you have 6 peas, what is the probability of having 6 yellow peas, 6 green peas, and everything in between, 5 yellow and 1 green, and etc. So and this is exactly the same way to go about it as I just told you. It's just a bit longer. You look at the Pascal coefficients for 6 events, as I just showed you in the previous table. These coefficients are 1, 6, 15, 20, 15, 6, and 1. Underline here, okay? And then you develop your terms. So we start with P6, P5, Q1, P4, Q2, etc. The powers of P and the powers of Q always equal 6, which is the number of events that we're considering. You include the coefficients. So it's P6 plus 6 P5Q plus 15 P4Q2, etc., etc. This is all equals to 1. So you add all these events. And here you have all your events with all the probabilities listed for you. For example, this 15 P4Q2 is a probability of 4 yellow and 2 green seeds in the pod. Namely, it's 15 times 3/4 to the power 4 and 1/4 squared. Remember, P and Q have different values. This 20 P cubed Q cube is 3 of each. And here you have 6 PQ to the power 5. That's 1 yellow and 5 green seeds. So here are all our possibilities, as I just showed you. Here are all the Pascal coefficients that that I just listed for you. And the powers of P and Q with the coefficients, and this is equal to 1. And these are the numbered probabilities given that P is 3/4 and Q is 1/4. So you'll note one thing here is the probability of 5 yellow seeds is 17.8%. The probability of, 6 yellow seeds is 17.8%. The probability of 6 green seeds is 0.02%. So you see, it's a lot lower probability of having 6 green seed compared to 6 yellow seeds. Why is that? Well, evidently because the probability of a yellow seed is much higher. Okay, it's 3 times higher than the probability of getting a green seed. So you see, the probabilities are skewed towards the number of yellow seeds in this case. Whereas for boys and girls, the probabilities were equal, no matter what the whether you're considering boys and girls in their numbers. Any questions? I can pause here for a minute, see if someone wants to ask something. Okay. But anyway, let me conclude here. I mean, I think this is kind of a fun little thing to think about, and it applies to many situations, but mostly in genetics, you know, it's actually quite important to remember that and to know how to do that. Now, let me just, before we go into pedigrees and recessive and dominant traits, which is something, you know, I think we can go very quickly over that. I want to mention these fun little factoids that you have in this table at the end of the chapter, because in fact, we know now the molecular determinants of these phenotypes that Mendel described. I just want to show you this. It's really interesting because it bridges the molecular biology that we've done earlier with the transmission genetics as it was described by Mendel in the 1860s. For example, that seed shape that we talked about, round and wrinkled, we now know that it's a mutation in a starch branching enzyme called amylase, and the mutant allele contains an inserted segment of 800 base pairs in length, and this is called a transposon. So it's a transposition event that inserted into the gene and inactivated it completely. So we know really what really caused that phenotype at the molecular level. And I'm going to let you look at that table. I just want to tell you about seed color and flower color because we talked about it before last time with the crosses. And so the yellow color has to do with which is the dominant, the wild type, has to do with an enzyme that breaks down chlorophyll. So it changes the seed at some point, changes color from green to yellow because an enzyme degrades chlorophyll during maturation. And this is a mutant allele in the gene that encodes that enzyme that contains 2 base substitutions and a base pair insertion that completely inactivates the protein that's made out of that gene. The flower color, remember the wild type is purple and the mutant is white. Again, you have, so this is a mutation in a transcription factor called BHLH. This stands for helix loop helix, which is a structural motif that's commonly found in transcription factors. And the mutant allele contains a base substitution that leads to a defect in the protein made out of that gene, and that inactivates that transcription factor. So this is just to conclude a little bit, if you will, on you know, what we know now regarding these mutations. Okay, so now let's switch gears a little bit and move to something highly related, but you know, to apply a little bit what we've learned with Mendel, and that's pedigrees. And I want to talk about dominant and recessive traits. They don't necessarily have to be diseases or mutations. I mean, we often call that diseases, but I mean, it doesn't have to be a defect. So if you don't know this, you have to take a peek at this. These are the symbols used in pedigrees. Female are symbolized with a circle and males with a square. And the filled circle or squares are individuals in a pedigree that express the trait. And another thing I want to mention is each generation is labeled with a Roman numeral, and in each generation from left to right, the individuals in a generation are labeled with Arabic numerals. And so individual, say, 2.3 is in the second generation, the third individual from the left. Now, look at this pedigree, and what you see here are a number of characteristics that I listed here and that are also listed in the table in the next slide. You see essentially that you have an affected individual. I say affected, again, take it as bit as a neutral assessment here. I mean, it could be hair color, it could be shape of the nose, or it can be anything, it can be a disease too. But you see, I have an affected individual at each generation. And so it's apparent, between quotes, vertical transmission. The trait appears in each generation. Each affected individual has an affected parent. Overall, one affected individual can transmit to half the offspring. And I say on average, sometimes it can be more, like in this generation here. Well, this is the two parents are affected. That's not a good example, but here's another one where all the kids are affected. But it's like overall in the pedigree, if you count the affected individual and how many affected parents they have, overall it's 50%. Can be more, can be less by chance. Two unaffected parents do not transmit the trait. You have this case over here and this case over there. And two affected parents may have unaffected children. You see that with individual 4, 10. So all of this is something that will clue you into autosomal dominant inheritance. We're going to talk about sex chromosome and inheritance at the end of this lecture, okay? Sex-linked inheritance. So all of this is clearly autosomal inheritance, and that is you have a single copy of the gene that provides the trait, and there's a 50-50 chance of transmitting the trait to the progeny. And if that gene chromosome is transmitted, then you see the trait, and 50% of the time it's not, so you don't see the trait. Okay, so this is autosomal dominant trait. Here's a table essentially telling you the same thing. Males and females have a trait in about equal frequency. That's the point that it's not sex-linked inheritance. We're going to see what happens with sex-linked inheritance at the end, as I said. And etc. This is essentially repeating what I just told you in the previous slide. Here's another case. You see, very different. You see that the trait can appear like this from unaffected parents. And this is called, in between quotes, horizontal transmission. The trait can appear at once in a given generation, and it can appear among siblings. I don't think horizontal is a fabulous term for it, but this is what it's often sometimes referred to. Affected individuals can have unaffected parents, and you can see that here and over there. If both parents are affected, all children are. This is this couple here that has 4 kids, they're all affected. And there's an observable effect of relatedness. What does that mean? It means if there is some kind of family relationship, even if it's somewhat distant, there's a much higher probability for recessive trait or recessive disease um, to in fact be transmitted as 2 recessive alleles. In other words, the chance that both members of the couple are heterozygous is a lot higher than if you have one person that's heterozygous and the other one from the general population. So to have sort of a relatedness in the formation of the couple uh, highly increases the probability of transmitting a recessive disease. So here's again the table sort of from the book that repeats a bit what I just told you. Keep in mind that males and females in the previous pedigree with the trait are approximately equally frequent, so it's not sex-linked. That's another point that I'm making now. You'll see that's not the same when we talk about sex linkage. I'm going to see that very soon. Now, just a quick word here. This is not a big deal, but this is back to like this conditional probability that I was telling you about, where you actually can guess what's going on in the previous generation based on the progeny. So simply put, if you have one affected individual here and 2 unaffected parents, clearly they're both heterozygous. Okay, so this is very clear. But if you go back, you know, what is the chance of having this woman be heterozygous, knowing that her brother is heterozygous? And what is the chance of having say this individual or that individual be heterozygous? So you know this is where this is why it was telling you conditional probabilities are a lot more complicated than what you know you have in the book. It really deals with complex pedigrees and sometimes a single event that can allow you to ascribe a probability on another individual to have at least a copy of the gene that can be fairly distant in relations, in family relation to that individual. But I'm not going to insist on that. This is very simple. You can clearly see 1 and 2 are heterozygous, and they transmit the trait 25% of the time. Any question or comment before I go to the next topic? Karina.
So when there is no sort of gender bias observed, then we can assume it's autosomal, or?
Yes.
Okay, thank you. So it's pretty clear when it's X-linked or sex-linked.
Yes, it is, and you will see that in a few minutes.
Great, thank you.
Yeah, we're going to talk about sex linkage now. Autosomal recessive is not sex-linked. No, we are distinguishing the autosomes, the non-sex chromosomes, with genes that are part of X chromosomes mostly and Y chromosomes as well. So we're going to talk about this in a few minutes. And speaking of sex linkage, well, here's our friend the fly. And you know that there's a gene already because I talked about it, the white gene, that's on the X. So we were going to analyze the transmission of the white gene. We talked about the white gene when we discussed variegation, if you remember. And now we're going to look at how this gene segregates. Here's the phenotype. This is the wild type eye color, this very beautiful compound eye that's has this nice red color. And this is the recessive mutant, white. And the gene that's mutated is called white. So white plus is red. I know this is a bit confusing, but the white gene, when it's wild type, imparts the wild type phenotype, so it's a red color. And when white is mutated, you have a white eye. Okay, so there's a gene in flies called white. It leads to white eye color. Okay, so let's look at a cross. First, we're going to look at flies, and then we're going to look at chromosomes, meaning we're going to have Punnett squares. So I'm going to show you this slide twice. First, here's a cross between a wild-type female and a male with white eyes. You do that cross, all the progeny F1 has red eyes. So right off the bat here, what you know, the mutation is recessive. You do sibling crosses and you get an F2 with, first of all, out of 4 possibilities, only the half the males have white eyes. So the only time you recover white eyes is on males, and it's only on half the males. Okay, so all the females have red eyes, and of the males, half have the wild type phenotype and half have the white eye. So you can immediately see here an imbalance in the recovery of the phenotype in terms of male versus female. So this is already an indication it's sex-linked. Here's another cross where, in fact, it's the reciprocal cross, where you have females with white eyes and males with red eyes. And here, what happens in the F1? The females are wild type, and all the males have white eyes. Again, we have a complete imbalance in the display of the phenotype, because only the males display the phenotype. That's evidence of sex linkage. See, very different from the pedigrees I just showed you. You do a sibling cross here of F1 between females with red eyes and males with white eyes, and then there you get a 50-50 F2. Half the females have the phenotype and half the males have the phenotype, so it's a 1-to-1 ratio. So in the reciprocal cross, all the males have the white eyes in the F1, and 50% of males and females have white eyes in the F2. But again, in the F1 of cross B and the F2 of cross A, there is a strong imbalance in the fact that the males recover the phenotype, and therefore it's an indication of sex linkage. Here are the same crosses, but now we have what happens at the level of the chromosomes. And what you can see is that we can explain these results by the white being present on the X chromosome, and the males being XY, since they have only one X chromosome, will display the phenotype if they receive an X chromosome with a mutation, whereas the female being having 2 X chromosomes, of course, will only display the phenotype if they receive the mutated allele from both parents. Okay, so it's a much rarer occurrence. So here's the first cross where we have the males with the white eyes, and the gametes that are produced to do the Punnett square are the Y chromosome, of course, which doesn't have the gene, and an X. Sorry, I apologize. I meant to say, first of all, you need an F1. So you're producing a female that is heterozygous for the white locus. That female in the F1 receives a wild-type copy from the parental female and a mutant copy of the white from the male, so they're heterozygous for the X. So that female's gametes will be 50% white plus and 50% white minus. Whereas the male, the reason why the males have red eyes in the F1 is because the males receive one of the X chromosome from the parental female and the Y from the male. So all the males have red eyes. They have all the wild type copy of the gene received maternally from the parental generation. So the gametes of that male will be Y or the wild type copy of the gene. And so this is the Punnett square that's produced. And all the females have at least one copy of the wild type copy, at least one wild type copy of the gene. So they all have red eyes, whereas the males have the Y, and 50% of the male will receive the mutant copy from the F1 female. Okay, so this Punnett square explains the result observed in cross A. So in other words, the reason why only the males have white eyes is because they only have one chromosome that's received from a heterozygous female in the F1. So 50-50 white eyes and red eyes. Let's look at cross B. So once again, you get an F1 from the reciprocal cross. So in that case, the females in the F1 are also heterozygous because they receive the wild type copy of the gene paternally from the parent generation. And the males have all white eyes because they receive, no matter what X they receive from maternally, since the female has white eyes, it'll have a mutant copy of the gene, mutant allele. So the male transmit the Y, but the X that they have will necessarily be white minus. And the females is 50-50 white plus, white minus, as in the previous cross. And so for that reason, 50% of the females will have white eyes and 50% of the males will have white eyes. So in fact, the phenotype in that case will be transmitted equally to both generations in that particular case. But you see that you have an imbalance in the F1 towards the males in this cross. Females never display the phenotype. And in cross A, again, you only recover the wide eye phenotype in the males. So this is indicative of sex linkage. Okay, so the Y gene is on the X chromosome. The males are so-called hemizygous for the X. That is, they only have one copy of that chromosome. This is a technical term, so it's not homozygous, it's not heterozygous, it's hemizygous because there's only one, the other chromosome being the Y in males. The females have two X chromosomes and they will have white eyes only if they are homozygous for the white mutations, as occurs 50% of the time in cross B in the F2. The white mutation is recessive. At the heterozygous state in females, the eyes are red, and so therefore they have the wild type phenotype. This is just a remark to tell you that this was actually a bit more complicated to do. This was done by Thomas Morgan at Columbia University. So all of this work and the isolation of the white mutation, the analysis of the white mutation, and the determination of sex linkage was done in New York City at Columbia University in Thomas Morgan's lab in the early 1900s, like between 1910 and 1920. And what I want to say here is is that the wild type females from that are used. Let me go back. So you see that in that case, in cross A, some of the you have to use females that are heterozygous, and this cross in the F2 only works if the females are heterozygous, and yet they have a wild type phenotype. If the female is homozygous, then all the progeny has red eyes, no matter what the phenotype of the parental male is. And so this was something that was analyzed very rigorously in Thomas Morgan's lab, a little bit like Mendel when he verified whether heterozygous, homozygous. Now, in Mendel's case, he could do it because he could self-fertilize the plant. In Thomas Morgan's case, it was a bit more difficult because you have to cross two animals. And so all of this was done by doing hundreds of crosses. And in 50% of the crosses, you had female progeny that was 50% white, white eyes and 50% red eyes, but only in half the crosses. So this was sort of a large number experiment requiring many crosses, because some of the times, without knowing, they were using a female that was homozygous for white, and in some other crosses, the female was heterozygous. That was just a point in passing. It's not all that important. It's really important to understand the sex linkage here and the Punnett squares. This is really the important part. Now, here I'm going to try to go fairly slowly because it's a difficult slide. I'm warning you, and you're going to have to spend some time on this, okay? Because this is something that requires your understanding of meiosis. So you're going to have to make sure you understand meiosis 1 and meiosis 2 and how they occur. And at the same time, superimposed to that, you have the notion that sex determination in flies occurs in a specific manner, and you have to remember that. And this is what I wrote here on the right side of this slide. This is how, in Thomas Morgan's lab, specifically by his graduate student called Calvin Bridges, this is how the first mistakes in meiosis were discovered. And these mistakes in meiosis are very rare events that result from chromosome missegregation. And this chromosome missegregation can occur during meiosis 1 or during meiosis 2. In that particular case, we have a mistake. Again, a very rare event. It's only in 1 in 2,000 flies in the cross that I'm going to tell you about now. It's very rare. It's a chromosome missegregation that occurs in meiosis 1. So let me tell you how he discovered that. Here's a cross. Look at the parental cross. So you have females with white eyes and males with red eyes, which was cross B, if you remember, in a few slides ago. Okay, so what happens if you do that? Okay, what I just told you is, okay, all the females are heterozygous. They received a wild type copy of white from the male and a mutant copy of white from the female. So all the females have have red eyes, right? All the females have red eyes, and they're all heterozygous. And then the males, they all received either mutant copy of white from the parental female. So all the males have white eyes. This is really what is this cross here. If you go back, that's the cross, see? So all the females in the F1 have red eyes and all the males have white eyes. Well, very rarely, Kelvin Bridges noticed in this cross that in fact he had white-eyed females and red-eyed males at the same frequency. How can that be? It's not supposed to happen. Well, this was due to chromosome non-disjunction in meiosis. So I'm going to explain to you how that works. But before, just a word on sex determination in flies. You know already that in flies, as in humans, XX is female and XY is male. But as I told you also, because remember we talked about sex determination with sex lethal and sisterless, the molecular uh, uh, events that occur are very, very different in flies. Very different. So this similarity is only superficial. So here's what you have to remember. And look at what I wrote here. You see that, and you'll be okay. Turns out in flies, XXY is female. That's not true in humans. In humans, XXY is male. But in flies, it's female because, in fact, it's the number of X relative to the autosomes that counts. So it's different. And so in flies, XXY is female. And in flies, X0 is male. Also different for us humans. X0 is actually female in humans. So I don't want you to be confused. In case you knew that, make sure you remember in flies it's different. Otherwise, you're not going to understand this. So once again, in flies, XXY is female and X0 is male. So you understand how in this cross, the great majority of progeny are red-eyed females and white-eyed males. But here's what can happen in one in a few thousand meiotic events. And this can happen in the male or the female germline, but in this particular case, we're going to consider the female germline. There can be a chromosome non-disjunction in meiosis 1, meaning the homologs, as you know, they pair in meiosis 1. And they go to the metaphase plate and they separate in anaphase. Well, very rarely, there's non-disjunction, meaning the two homologs go together in meiosis 1 in anaphase, and that gives you one myocytes without X chromosomes, if we consider the X, and another one as a result of meiosis 1 with an extra X chromosome. So this is called an aneuploidy for the X chromosome. So you're going to get, as a result, after meiosis 2, out of 4, 2 gametes that have no X chromosomes. Now, what happens with the other chromosomes is normal, okay? This is a very rare event, so this very, you'll have everything going normally for the rest of the autosomes, except that it doesn't have any sex chromosomes, 0X. And on the other side, you'll have one cell, myocyte, after meiosis 1 that has an extra X chromosome, and that extra chromosome is just going to line up at metaphase 2, just like a regular chromosome, and the sister chromatids are going to separate, and you're going to get a gamete with an extra X chromosome. So a non-disjunction in meiosis 1 gives you 2 resulting gametes without a chromosome that are aneuploid -1 and 2 gametes that have a supernumerary chromosome, an aneuploid +1. So now let's think of what happens when these gametes are fertilized. The male produces a Y and X with a wild-type copy, as you know. It has red eyes. Let's first look at the gametes without a sex chromosome. This is an X0 with a white plus, because it's the X from the male. And you see that, as I told you, X0 is male, but it has a wild-type copy of the gene, so it's a male with red eyes. And funnily enough, it receives its X paternally. I say funny because normally males do not receive the X paternally. They receive the X maternally. They receive the Y paternally. So this is what happens, and reload. Y0 is lethal. It's not viable, so let's not consider this. So it's the Y with the aneuploid -1. You don't see it. And let's look at the one that's aneuploid +1. This getting fertilized by the one is an XXY, but the two X have a white mutation because they inherited maternally. Okay, they're just two different homologs, but as you know, the paternal situation here in the female is homozygous. So we have XX with homozygous white mutation with a Y, and XXY is female with white eyes. And this is how you're getting these rare events that in fact do not correspond to sort of the progeny that you expect. That is a result of a aneuploidy in the gametes due to chromosome non-disjunction in meiosis 1. You can have also chromosome non-disjunction in meiosis 2, and we're not going to cover that right now. We'll talk about it later. Any questions about that? You probably, I know you're going to have to look at this carefully. Email me or talk to me next week if that's a problem. Karina, you have a question?
Yes, so this they got on a tiny scale, I assume, right? It was literally
Yes.
one female fly, this one male fly, and they happened to have maybe a handful of these crazy mutant offspring.
It's 1 in 2,000 progeny flies. That's the frequency. See down there.
Yeah. Okay.
So it's not only rare, it's very rare, but also it's so rare that you really won't have more than one. It's like you can assume that the gametes produced by the male are going to be wild type, because some people say, well, this can also happen from the male, and then what happens if the fertilization occurs between two aneuploid gametes? The proportion of that, is that probability squared, the
Right.
product rule, right? So you can assume that these missegregation occur only on one side. They can occur in the male gametogenesis as well. Yeah.
Oh, I see. Thank you.
Okay. Yeah. Okay. Great. So yeah, like I said, be careful. This is a complex slide, I know, and it's like it, but I like it because in the sense you really need to understand meiosis to this. And let me just make one more comment. What happens in this aneuploid meiocyte is like the extra chromosome behaves just like it behaves just like the other chromosome. It's going to go independently at metaphase 2. It's just going to line up like the other chromosome, and the sisters are going to separate, and you're going to produce these two gametes with an extra chromosome with one chromatid. All right. Okay, I want to finish by talking about perhaps material that's a little bit less conceptual. And I just told you, the sex determination in flies and in mammals is different despite the apparent similarity in the sense that XY is male and XX is female. But in all cases, there's an issue, that is, there's an imbalance in the number of genes coming from the X. And there are mechanisms that compensate for that. They call dosage compensation to equalize gene expression for the X chromosome. And this dosage compensation takes very different mechanisms from organism to organism. And we're going to look at what happens in mammals. In mammals, one X chromosome is randomly inactivated in each female somatic cell. So we're reducing the gene expression by rendering one X chromosome randomly, either the paternal or the maternal one, where we're rendering heterochromatic. This happens during gestation, during development. So it's all set at birth. And at birth, the somatic cells, some will have gene expression coming from the maternal X chromosome, and some other cells will have gene expression from the paternal X chromosome. And flies and worms have different mechanisms to do that. So mammalian males and females have different number of X. There is a dosage compensation in female mammals, and that is done by random X inactivation early in development. One X is left fully active in each nucleus. The other X is almost entirely heterochromatic, and it forms a very small, dense body that can be viewed by a conventional light microscopy, which is a body called the bar body in female cells. The inactive X is largely rendered heterochromatic. So one of the X chromosome, the maternal or paternal, is randomly inactivated during development, and this inactivation is transcriptional, and it relies on an RNA called exist. I'm going to show you that in the next slide. And it's the X chromosome that transcribes the exist RNA that gets inactivated. More on that in the next slide. But you see, this is essentially what you see during development. You know, very quickly after fertilization, there is proliferation in to form the embryo. And then in some cells, you're going to have the paternal X being rendered heterochromatic, or the maternal one. And then there is formation of the full embryo, and essentially females can be viewed as mosaic for the X chromosome gene expression. Some cells express maternal copy, some cells express the maternal copy. So this is this thing with exist. It's a really fascinating mechanism. And exist is a locus on the X chromosome, and it is transcribed from both X chromosomes early on, before inactivation. And essentially, one X chromosome will keep making it and will shut off transcription of exist on the other X chromosome. And that chromosome essentially will acquire exist RNAs along the chromosome arms, and this exist RNA leads to silencing and condensation and heterochromatization of this X. So exist is a structural RNA. It doesn't make a protein. It's not translated. It's only transcribed. And one X chromosome essentially gets to make it and to shut itself down. And if one wins, the other one doesn't. And the other X chromosome is the chromosome that's going to be expressed the rest of the course for the cell lineage, if you will, after the cells have divided. This heterochromatization has to do with histone methyltransferases. So these are enzymes that methylate the histones, notably H3 and H4, as I sort of alluded to a couple of weeks ago. And that leads to histone deacetylation inactivating transcriptionally the whole chromosome. Now, one of my favorite slide. I told you females can show mosaicism if a phenotype can result from expressing one cells for one X or the other, and there's a locus on the X chromosome of cats, which is called the orange locus, that has different alleles, and you see that all the cells from these patches on this cat all originate from a single cell during development that has inactivated one of the X and left an allele that expresses the black pigment, and the descendant of these cells then form these patches on the other allele. So you have one allele that gives you the black color and another allele that gives you the orange color. So this, of course, give you this phenotype in females of the calico cat. And specialist actually told me this is technically called a tortoise cat, but same thing. It's due to X chromosome inactivation. Okay, now let me show you a few pedigrees here very quickly before I finish. Sex linkage also is seen in humans for certain traits. So here is an X-linked recessive trait. Male that's affected, and you see that in that case some of the daughters are carriers, like this one for example, and 50% of the time these carriers will have affected males on average. Okay, here's another carrier female, and one son is affected, one is not. So this is into the details. I'm asking you to look at these pedigrees and understand them. But you see, you have more males than females that have the recessive phenotype. Some females are heterozygous. They do not show the phenotype, but they can transmit the trait. And you see here again that be careful also. It's like, oh, the father transmits to the son. Well, that's not necessarily the case. In that case, of course, it's the mother, you see, that is heterozygous for the trait. But anyway, all this is compatible with sex linkage as we discussed it in the fly. You're looking at the Y gene. Here's another example with X-linked color blindness, well-known case. And here's another example of a recessive trait that's sex-linked, which is hemophilia A, with a carrier known as Queen of England, Queen Victoria, and that has essentially transmitted this trait, again X-linked, to some of her descendants in a number of different families. Can you have dominant traits that are X-linked? Answer, yes. There's a trait called dominant hypertrichosis. It's extra formation of hairs on the body. And so here's a female that's affected. Transmits 50%, vertical transmission, overall 50% transmission, as we defined it when we looked at the dominant traits. And in that case, you see there is an imbalance in say transmission from the male. You see clearly the males transmit to females. It's a dominant trait to all the daughters, but that father doesn't transmit to any of his sons. So again, you see this in that particular case in transmission from the father. You know, it's very clear that it's sex linked. finally, table showing some X-linked dominant and X-linked recessive traits. I'm gonna let you look at this if you're interested. And just a quick comment here. You see these OMIM numbers, OMIM, and I point that term at the bottom of the table, stands for Online Mendelian Inheritance of Man. You can Google for that. You get to the site, and it is a database of human genetic diseases. And you can search for the name of the disease, or you can search for the OMIM number, and you'll get a lot of information that relate to the symptoms, the medical, the treatments, what's known about the biochemistry and the molecular biology of a trait or a disease. So if you look at, for example, color blindness, and you look at this OMIM, though it'll tell you what genes are affected in that disease and why it leads to color blindness, what kind of mutations lead to this condition. You'll get all of what is known in terms of the medicine and the biology is actually listed. So if you're interested, you can look at that. Okay, this is done for what I wanted to say today. I'm going to stick a little longer, but if you have any questions, you're welcome to ask them. Otherwise, I wish you good evening and a good weekend, and I'll see you on Tuesday.
Professor, actually, I have a question.
Mm-hmm.
For the exam.
Yeah.
Are you gonna post like something like what's most important that we should know about?
I think I've done that. I post these topics for each slide. So yeah, I mean, I think it's also for you to.
No, I thought you were gonna post a review, exam review.
No.
Oh, okay.
I mean, to me, that's what the homework is. And many people have asked me, if you can expect the same level at the exam of what you see in the homework. So since you ask, I'm going to say, I've been asked that several times. In the homework, you have some questions that of what you can see in the exam. Those are like I think they're very straightforward, multiple choice, what's going on, and you have 4 choices and you pick an answer. So that is the kind of thing you're going to have at the exam. But in the homework, you'll have like questions that are a lot more complex from what you're going to see at the exam. You're not going to have multiple part questions, like you see in the homework where you need an answer and then you need to carry on with that answer. That I'm not going to do because I think it's too much. And I'm not going to have things that take too much time to answer. You're going to have a lot of questions to answer fast, and those questions will be destined to test you on whether you've seen something before and whether you can look at the answer quickly. Does that answer?
Thank you, Professor.
Okay, yeah, you're welcome. Okay, Karina, you have a question?
Yes, my question is kind of more general because now looking at that bar body,
Yep.
and the cat and all that, it makes me wonder, so we have 2 alleles of every gene. Does every cell in our body just pick which one it expresses? And then why aren't we mixes of things, Why aren't there patches of dark and light hair on my head or things like that?
Yeah, so in general, the X chromosome inactivation is to compensate for the difference in number of chromosomes between male and females. So see the X essentially, in principle, having a level of gene expression that is a level that's compatible with having 1 X and not 2. but that's not true for the autosomes. Everybody has two autosomes, two homologs, as we know, and so for these chromosomes, the level of gene expression is compatible with having two loci, two homologs of each chromosome. This being said, there is a mechanism for a few hundred genes, so not many, but there is a mechanism that inactivates selectively either the paternal copy or the maternal copy at a number of places in the genome. So that is on a number of different autosomes. That mechanism is called imprinting. And so for a few hundred genes, all of us, we express only the paternal inherited copy or the maternal inherited copy. So it's a mechanism that exists, but that is by and large, no, it's in small minority. Regarding patches in the body or of cells that seem different from others, that can be due to another process which is called mitotic recombination, where two homologs in a cell can recombine and form homozygosity out of heterozygous situation. So it's a totally different mechanism, but it does happen.
I see. Thank you. So generally,
Okay.
we do use both loci
Yes.
of our genes at all times. Okay.
Yes, generally, yes.
Thank you.
Yeah. Carolina.
Yeah, hi, Professor. I have
Hi.
two questions. So the first one is for sex-linked genes, like mutations and stuff like that, if there is an infected female offspring, she has to have an infected male parent, correct?
I believe so, yes, because that female must receive 1x from the father or the male and 1x for the female.
Okay, thank you. And then
Mutate with a mutant copy, yeah.
yeah. And then
Okay.
can you go back to autosomal recessive inheritance, and can you just quickly describe the horizontal transmission again, because I don't really understand.
This.
Yes.
Yeah, I mean, look, this is a situation where this is for a recessive gene. So an affected person by necessity will be homozygous for the mutation. And the other parent is wild type, so you can assume that they're homozygous wild type. So that parent, you see, this male will provide a wild type copy of the gene. So all of the kids will not be affected. Okay, but then in that case, okay, what's happening here is you happen to have two unaffected parents that are both heterozygous. And if you have two unaffected parents that are both heterozygous, the chance of a child inheriting the mutated copy of the gene is 25%. This is the product rule, by the way, because one parent giving the mutated copy to the kid is 50%, okay, and the same has to occur on the other parent. So and the mother has to give the mutated copy. That's 50%. Remember, and is the multiplication rule. So 0.5 times 0.5 equals 0.25, and that's 25% or a quarter of a chance of actually transmitting a disease from two carriers, unaffected parents.
Okay, so when you say trait can appear at once in siblings, what do you mean by that?
Yeah, this is not a fantastic pedigree to show that, but imagine this couple, okay? And we know they're carriers, right? Because imagine they have another 5 kids. Well, there's a chance of having 1 or 2 more siblings with the trait. So then, you'd get a family with more than one sibling showing the trait.
Okay. Thank
That's
you.
all. But you don't actually see that in this pedigree. So yeah.
Okay, thank you.
Okay, you're welcome. Okay, Maxime.
Yes, hi, Professor. I have a question.
Hello.
Is this going to be the last lecture for the test?
Yes,
And
the lecture on Tuesday will not be part of the test.
and also, will you be asking us any mathematical questions or like conditional or statistical to actually like figure out?
In the case of binomial probability, what I can do is do the math for you in an easy way. And if you understand it, you will immediately see the answer. I'm not going to ask you what's the chi-square for this cross because it involves, a calculation that's easy to jumble in a stressful situation if you have a minute to do it. But I think the questions I'm going to ask you will be, I wouldn't call them math questions. They will test your understanding. They will not test your ability to use a calculator quickly. I know it's stressful. I know it's a difficult circumstance. So I think I will not expect you to calculate a chi-square or a binomial probability. For example, if I go to this slide, right, I'm not going to ask you to calculate these numbers. But I will, for example, give you like different types of formulas for you as a multiple choice, and you will immediately check on the one that's correct.
Thank you so much.
Yes. Jason.
Hi, Professor. Yes.
Hi.
What kind of questions can we expect on your exam regarding interpreting pedigrees?
What you have on the slide. And it's going to be, again, something that's, if you review the slide, if you understand it, you'll come up with the answer very quickly.
Beautiful.
Nothing, no curveballs. I know with pedigrees, you can have curveballs. You have like a huge pedigree with like one individual that sort of shows you that it's something very no curveballs. Okay, no, I think I can say, and I hope you guys will agree with me. But this is not really to have something like exceptional that you have to sort out. It, nothing difficult.
Thank you.
As long as you understand what I said today, you understand the slides, you'll be okay.
Beautiful. Thank you.
Yeah. All right. Any other questions?
Professor, I
Yes?
had a question. You said something about the the one X chromosome inactivating the other X chromosome. So that's in case of us human beings. Like all the
Yeah.
females have triple X's, but one X inactivates the other X, and all the males have two X and one Y, and one X
No,
is
no, no,
responsible.
no, 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 no. Excuse me. No. Females have two X chromosomes.
Mm-hmm.
They're XX, and males have one. They're XY. And so in females, you have two X, and there is essentially there's a competition between the two, right, so to speak. And initially, both X's start to exist, but one wins over, probably because it's expressing more exist RNA. And the exist RNA will bind to the chromosome that's expressing the RNA itself. So this is one of the X chromosomes. It will inactivate itself, but this RNA expression will lead to the repression of exist transcription on the other X. So the resulting situation is one chromosome will make this RNA and it will inactivate itself. And the other chromosome will not make this RNA and will stay active transcription.
So only one of the X chromosome gets to show itself.
It gets to be
Express itself.
transcriptionally active. Gets to be transcriptionally active. Let's
So
say it that way.
So for this happens for like all females, that one of the X chromosome
In
will
all cells, yes.
all cells.
And
Okay.
in all cells, yes.
Okay then.
The males having only one X chromosome, this doesn't occur.
Okay then. Thank you, Professor.
By the way, since you're asking, you know
Mhm.
there's some males who are XXY because they had a non-disjunction in meiosis 1.
Correct.
And these are males in humans. Well, so there are males who have two X chromosomes, and they will have a bar body. So this is really something that occurs in males, but rarely because they need to be XXY.
Thank you, Professor.
Okay.
Professor,
One more question.
yes.
Nor.
All right. Oh,
Hi.
No,
okay.
Professor, that was me.
Oh.
Who asked the question already?
Oh, I'm sorry. I'm sorry. I saw your hand. That's why I wasn't sure it was you. Okay. I heard a voice.
Yes, that's me. So like,
Hi. Yeah.
when is the case when they refer to super female and super male?
I'm sorry. What's the question? I didn't hear very well.
Like, like sometimes like a female has like super female and super male. What does
What do you mean by that? I'm not sure what you mean by that.
it's like X, when a female have like 3X chromosome and then a male have like an extra Y chromosome.
Okay, the males with extra Y chromosome, they have some. Can you hear me?
Yes, I can.
Okay, I don't know. Something happened. Um, Yeah, so this is just a different situation. It has nothing to do with X chromosome inactivation. These are syndromes that occur through, in fact, meiotic non-disjunction, as we talked about in the fly. So some males can inherit an extra Y. And there are other characteristics where the sort of a chromosomal um, context is in fact normal, can be XX, but there's some other physiological issues that occur that have nothing to do with the number of X chromosomes. They're hormonal or things like that. So this is something that's a different issue.
All right, thank you.
Yeah. Okay, you're welcome. All right, so I'm going to stop here, and I wish you all a great weekend, and I'll see you guys on Tuesday. Bye, everyone.